Good morning, students. Today we are going to discuss on topic bacterial transduction. In semester 2 unit 4 we have bacterial recombination methods and mechanism. So bacterial transduction is one important bacterial recombination mechanism. Today we are going to understand the basics and applied aspects of bacterial transduction. Definition of transduction is the transfer of DNA from one bacteria to another bacteria with the help of bacteriophages. We have studied about viruses. There are three different viruses: plant viruses, animal viruses, and bacteriophages. So bacteriophages are the viruses which can attack specifically bacterial cells. These viruses, as we all know, have two important components. One is genetic material, it can be DNA or RNA, and they have a protein coat surrounding the genetic material. Bacteriophages can replicate by two modes. One is the lytic mode, the other one is the lysogenic mode. This we can easily understand with the help of a diagram. In this diagram, on the left side you are finding the lytic cycle. On the right side you are finding the lysogenic. Lytic cycle is mostly followed by the virulent phages as a part of their life cycle, while the temperate phages can follow both the lytic as well as the lysogenic. Now the basic steps in the lytic cycle: first, the bacteriophage adsorbs on the bacterial cell membrane or cell wall. This site where it adsorbs or attaches, it is a receptor. Now after it adsorbs, then it will try to inject the phage genome or viral DNA. So this step is called as penetration step. The viral DNA penetrates into the cytosol of the bacterial cell. After the penetration, now the viral DNA can easily undergo replication. During this process, only the viral DNA is allowed to replicate, and the host genome or host DNA, which is now in this diagram you are finding it as a blue colored line, is hydrolyzed. It's cut into small pieces. Only the virus is able to replicate, and that results in a number of phage particles or virons. Because the number of phage particles are increasing in the host cell, that ruptures or lyses the cell and releases all these new particles. So first stage is adsorption, second stage is penetration, third stage is multiplication, and fourth is the cell lysis or rupture, which releases new viron particles, which again they can infect and start the cycle. So this is the lytic cycle. In contrast to this is the lysogenic cycle, where you don't have any multiplication of the phage particles, but after the viral DNA is injected into the host cell, it is getting inserted. So this insertion is very, very important, and because of this the viral DNA is becoming inactive. In this state it can stay for a long time. So the prophage is the name which is often used to understand this stage of the viral DNA. Now whenever there is favorable conditions, a prophage can easily come out of the host chromosome or get excised, and again it will enter into the lytic mode. I think this has given us a clear idea about the two different life cycles of the bacteriophages. Now the process of transduction generally is understood as two modes. One is called as generalized, the second one is a specialized. The generalized, as the term indicates, any portion of the bacterial chromosome can be picked up by the viruses and they get transferred to the another bacterial cell. So this type is called as generalized transduction. In contrast, the specialized transduction mode, a specific region of the bacteria is being identified and that is being picked up and it is being delivered to another bacterial cell. So this is called as specialized transduction. Now we will understand one by one. First we will move on how the generalized transduction is being discovered. Here two strains of Salmonella typhimurium are used, which have different genotypes as well as the phenotypes. The LA22 and LA2. These are two different types of the Salmonella typhimurium strains. La22 has phenylalanine defective, it can't synthesize. Similarly, it can't synthesize tryptophan, TRP, but it has the capability of synthesizing methionine and histidine. All the four are amino acids. La2 strain, in contrast, it can synthesize phenylalanine on its own. It can also synthesize tryptophan, but it lacks the necessary enzymes of methionine and histidine. So that's how we are putting as phenylalanine minus, tryptophan minus in case of La22, and this same two characters are plus. La2 can make this. So when I mix these two strains as per the recombination mechanism, I expect if both the parental characters are found in the progeny, then I understand the progeny is coming as a recombinant. Now how will I score them on the plate? I will take a plate which lacks all the four amino acids, put the mixture and find is there any colony is being growing in the plate. Now before I understand, I can also do one more experiment, that is U-tube experiment. In this U-tube experiment, I am taking La22 on one side of the U-tube and the La2 in the other arm of the U-tube, and both strains have been separated by a filter, porous filter. Now when these two strains are taken in two arms, there can be some sort of recombination. Now because of the recombination, when I grow the mixture onto the plates which are lacking amino acids, you can find colonies still growing because of the recombination. So how that recombination is happening, we'll be wondering. Now what is happening during this is La2 strain is a prophage. What do I mean by prophage? Just now I have explained. A prophage is a bacterial cell which is having the viral DNA in it. So La2 strain, if it is a prophage, when it is converted into a lytic mode, then some of the viral particles, they will be carrying the bacterial genes, that is the meth, phenylalanine and tryptophan characters along with them, and they will be injecting into La22 strain. So because of this, there is a transfer of bacterial genes from La2 to La22, and that is the reason why we are finding the recombination happening and the colonies are growing on plates which are lacking all four amino acids. So this is how the generalized transduction has been discovered. So now we will understand the mechanism of generalized transduction. Again, I am taking the help of a diagram where I am taking two examples. One phage, that is P22, which is known to infect Salmonella typhimurium, and P1, which is known to infect E. coli. So P22 will be our Salmonella will be our donor, and P1 can be our E. coli can be our recipient. So we can this generalized transduction is exclusively studied taking P22 and P1. We can find n number of examples in the literature. So first I am talking about Salmonella typhimurium. Assume this is our Salmonella typhimurium strain. Now when P22 infects, the Salmonella is assumed to have two characters, histidine and lysine markers. Histidine plus indicates it is capable of synthesizing histidine. Lysine plus indicates it is capable of synthesizing lysine. So when the phage infects the bacterial cell, as I have explained, the host DNA is lysed, hydrolyzed into small pieces, and the viral DNA replicates. Now a portion of the when the viral DNA is getting packed, instead of packing the viral DNA, by mistake, sometimes the bacterial genes are packed into the viral coat. So this is how a transductant is being generated. This happens very rarely, but it happens. So occasionally, a phage particle will be carrying the bacterial chromosomal genes or bacterial genes. So here in this diagram we can see that his marker is being carried by one of the phage particles. So this phage particle, I call it as transductant. So randomly some genes are picked up. So here for our understanding we are considering his marker is being picked up by this particle. So now this is the transductant, and when this transductant infects the recipient cell, that is another bacterial cell, when this is injected, when this his marker is injected, if the recipient is having his minus, so these two sequences, because of the homology in sequences of DNA, it will undergo homologous recombination, and because of the homologous recombination, the recipient bacteria now will become his plus. So because of the generalized transduction, we are able to understand how a recipient which was his minus has become to his plus. Phenotypical change is observed. So I repeat again, phage, once it infects the donor, picks up some of the bacterial genes, then you will be finding what is called as a generalized transduction is made, and this transductant, when it infects another cell which is lacking that marker, when it is injected, there you can find what is called as homologous recombination. So the transduced DNA is recombined into the chromosome of the recipient cell. So because of this, you can find the recipient cell is changed. So this is how the generalized transduction mechanism operates. Now, in contrast to this, we have other method called specialized transduction. In case of specialized transduction, only a portion, certain region of the chromosome is normally used for the transfer. So example, if I take lambda phage, which is known to infect E. coli, it cannot move all the genes. It will move only either GAL or biotin gene. So that's how specialized transduction cannot be used for general genetic mapping experiments. First, with the help of diagrams, we'll try to understand how the specialized transduction works out. So here is the diagram where blue color line symbolically represents the circularized phage chromosome with an ATT site. ATT stands for attachment. Then we have a bacterial chromosome which is having attached site for integration of the phage, and also on either sides of this attached site we are having a GAL marker and a biomarker. So when the integration happens, because of the homology between the two regions, you can find the lambda chromosome or the viral chromosome is inserted into the bacterial chromosome. This is a normal procedure of making a lysogen. So after the lysogen, when it is induced to lytic mode, then will be an outlooping where the phage genome or chromosome is again excised from the bacterial chromosome. This is a normal procedure, but rarely or in an abnormal way, a portion of the bacterial gene is being incorporated into the viral chromosome. In such cases, we will get what is called as DGAL phage. Now DGAL, D stands for defective, GAL marker is incorporated into the phage. So that's how I'm calling this as DGAL transductant or DGAL phage chromosome. Now this viral DNA is carrying the bacterial DNA, bacterial gene GAL+. So in this diagram you can find there are two types of transductants. One is called as lysogenic transductant, which is happening where you can find the double lysogens are generated. So 2 times the phage DNA is being incorporated into the bacterial. So when such things happen, so that goes with a high frequency of transduction. So such lysates, I call it as high frequency of transduction. In the second one, mostly in case of second one, you can find the GAL marker is again undergoing, this is the transductant. When it injects into the recipient cell, there will be what is called as crossing over. So exchange of GAL+ marker with a GAL- will result, and that's how a stable transduction happens. So at the end of specialized transduction, the Gall marker which is there on the transductant is undergoing recombination in the recipient. So a recipient bacteria which was earlier Gall minus will become Gall plus. So a stable